This is the first video of the ionotropic receptors first lecture. In this lecture, we're going to discuss the structural and functional properties of ionotropic receptors, also called ligand-gated ion channels. And in this first video, we're going to talk about the introduction, different types of synapses, neurotransmitters, types of receptors, and neuromodulation. So, as many of you know already, neurons communicate with each other via synapses. And there are two main types of synapses. There are electrical synapses and chemical synapses. And electrical synapses contain a large number of gap junction channels that communicate the pre- and postsynaptic cell. When an action potential arrives to the presynaptic terminal, the electrical signal travels through the gap junction channel, inducing an inward current in the postsynaptic cell, and therefore causing a depolarization of the postsynaptic cell. This type of synapses can actually be bidirectional. In contrast, in the chemical synapses, when an action potential arrives to the presynaptic terminal, it triggers the fusion of vesicles containing neurotransmitters with the membrane of the presynaptic terminal, releasing neurotransmitters to the extracellular space. And then neurotransmitters bind to specific ionotropic receptors present in the postsynaptic terminal. And activation of these receptors leads to changes in the membrane potential of the postsynaptic cell. In this case, an electrical signal is transduced into a chemical signal that is then transformed back into an electrical signal via activation of the ionotropic receptors. And these synapses are unidirectional. So for the next 3 lectures, we will focus on studying chemical synapses, specifically how different ionotropic receptors work and transform chemical signals from neurotransmitters into electrical signals. So let's start with what are neurotransmitters and what kind of neurotransmitters we can find in the brain. Well, neurotransmitters are bodies' chemical messengers composed by simple molecules, derivations of amino acids, or even small peptides that are packaged and stored locally into small vesicles. This type of amino acids are stored in vesicles that are around 40 or 50 nanometers. The first neurotransmitter to be discovered was a small molecule called acetylcholine. It plays a major role in the peripheral nervous system and have roles in maintaining and modulation both motor and cognitive function. Glutamate, on the other side, is an amino acid with a primary excitatory function. Conversely, a major inhibitory neurotransmitter is the gamma-aminobutyric acid, or GABA. And another inhibitory neurotransmitter is the amino acid called glycine. Many other neuromodulators are monoamines, including catecholamines such as epinephrine or adrenaline and noradrenaline. These are monoamines and work on the activity of various organs in the body to control things like blood pressure or heart rate, liver function, and many other functions. And then we can also find in this group dopamine, which is involved in many functions, including motor control, reward, and reinforcement and motivation. The neurons that use serotonin, which is another monoamine, functions in sleep or memory, appetite, mood, and others. Then another neurotransmitter from this group is histamine, which is the last of the major monoamines and plays a role in metabolism, temperature control, regulating various hormones, and controlling the sleep-wake cycle, among other functions. And then on the other hand, we have neuropeptides, which are small protein-like molecules or peptides found in larger vesicles, as we can see here. There are about more than 100 nanometers dense core vesicles. And few examples of neuropeptides are enkephalin, which is a pentapeptide involved in regulating nociception in the body, or somatostatin, which is a peptide hormone that regulates the endocrine system. So, important things are the many neuropeptides are also co-released with other small molecule neurotransmitters. And one major difference is that peptides are not recycled back into the cell once they're secreted, unlike many conventional neurotransmitters such as glutamate or dopamine or serotonin. And another difference is that after secretion, peptides are modified by extracellular peptidases, activating them or sometimes inactivating their function. So in this slide, there are different structures of some neurotransmitters that we just discussed. You do not need to know their structures. This is just something to illustrate how they look like. And I want to emphasize 2 points before we move on. One is that a neuron can release more than one neurotransmitter, all right? So there are not specific neurons that release a specific or one unique neurotransmitter, but they can release different neurotransmitters. And they're certainly ineffective without the appropriate postsynaptic receptor. If there's not a receptor that they can interact with, these neurotransmitters are useless. So there are 2 types of neurotransmitter receptors at the cell surface. We have ionotropic receptors or metabotropic receptors. And very briefly, because we will see more about the differences between these 2 types, it's just that ionotropic receptors here are ligand-gated ion channels and represent a direct and fast response. So upon neurotransmitter binding, the channel opens here and the ions flow. And the types of ions that can flow include potassium, sodium, calcium, and even chloride, and they can pass through the membrane. On the other hand, a metabotropic receptor here involves a complex of proteins that result in metabolic changes within the cell, producing a second messenger, and that acts from the inside to open an ion channel or develop an indirect activation or a cascade of other metabolic changes. Therefore, metabotropic receptors are involved in an indirect activation and are related to a slow synaptic transmission. And we will talk a little bit more about these metabotropic receptors in the next video. So these ionotropic receptors are proteins that are typically composed of at least 2 major domains. There is a transmembrane domain, which includes the ion pore, and then an extracellular domain, which includes the ligand binding location. And different types of receptor subunits may have additional domains, like in this case. And in this panel in particular, we can see a schematic representation of the general structure of a I-GLUR subunit of the ionotropic glutamate receptor, indicating the extracellular located amino terminal domain, or ATD, and a ligand binding domain here. You can see it better in this representation. And the transmembrane domain, TMD, comprised of 3 membrane spanning helices, together with a reentrant P loop here. This is just a unique characteristic of this subunit. And in response to the binding of a neurotransmitter, A resulting conformational change opens the ion channels, like we can see in this diagram, and leads to the flow of ions across the membrane. So the opening and activity of these receptors can be also modulated with different drugs, whose specialized functions in the recognition of intercellular molecular signals. That is, they act at the recognition site for neurotransmitters and hormones. So in this diagram, we can see how agonist drugs enhances the action of the receptor and can imitate the action of the neurotransmitter in the bid to enhance them. So agonist drugs can bind and act directly or indirectly. For instance, in the indirect binding, in the direct binding, the agonist drug works the same way as the neurotransmitter, and they bind themselves to the site of the receptor, as it is suggested in this picture. And a classic example of these direct binding agonist drugs is nicotine, which binds to the nicotinic acetylcholine receptors. Then the indirect acting agonist consists in that the agonist drug enhances the actions that the neurotransmitter have on the brain by encouraging and stimulating release of them, basically upping the amount which are released. So, on the other hand, antagonist drugs obstruct the path of the neurotransmitter here to stop them, and they can do so also directly or indirectly. And the last thing about drug neuromodulation is that the consequence of a drug receptor interaction is a physiological change whose magnitude can be measured. And here this is an example of how we can measure the response of a receptor at different drug concentrations. And in this last slide, you can find a summary of what I've just explained about the differences between agonist and antagonist. And finally, as usual, I leave you with a short quiz where you can revise some of the most important concepts that were covered in this introductory video.